COVID-19 and SARS-CoV-2 data. And so more broadly, though, I want to talk about what it means to share data in the midst of a pandemic. When I was asked to first give a title for this talk, I sort of jokingly gave the title of Why the Boring Stuff Matters. But at the end, I really want to return to the idea of the importance of standardization. I think it's essential when you're talking about sharing data in general, but when you're talking about time-sensitive data and analysis that needs to happen rapidly in the midst of the pandemic, it becomes critical. And so to start with, I wanted to talk about where we were a year ago when we really started this project. And so I think, one of the most striking things to me about the COVID-19 pandemic, especially in the early stages, was just the unprecedented level of sharing of data. So basically, as soon as data had been generated, there was research that was coming out to the minute, which opens up huge potential for collaborations and for all of us to take advantage of the shared knowledge that we're rapidly generating. But the challenge and the problem with that too is that it becomes increasingly difficult to be able to find what you're looking for and to try to make sense of all this information. And so this is a slide I put together in mid-January where I was just trying to find all of these SARS-CoV-2 virus sequences that existed across the web. And so the good news is that there's tons of information, but the bad news is that it's kind of scattered everywhere. And so the initial sequence was originally deposited in places like virological.org, which is a messaging board where people talk about viruses. People would talk about new data being released over Twitter. And then there are more standardized repositories for this information like GenBank or NextStrain, which is an aggregator which takes in all this information and then does analysis on it. So there's huge potential to take advantage of this data, but finding it is increasingly a problem. So that was a year ago, and the question is what has changed? And so I think the first thing is that, at least in the genomics world, there's been some consolidation in terms of where people are sharing sequences. So right now on GISAID, there are over 560,000 separate unique sequences that have been submitted, and this comes from over 1,000 unique different submitters. So there's a lot of information that exists, and it's starting to be consolidated into a few places. But the problem is that that's one type of data, and that's only data sets. And there's a lot of other information that's being generated about COVID-19 and SARS-CoV-2 as well. So in addition to data sets, there's also publications, there's loads of clinical trials, there's protocols for how to deal with the virus and the disease, as well as other things. And so when we created Outbreak.info in February of last year, we thought that there was a lot of information and someone should try to make sense of all of this. And of course, in the past year, it's become even more complex as there's been more diverse and heterogeneous types of data that are being shared. And so in this histogram of the number of COVID-19 stuff that we're tracking over time, since basically April of last year, there have been 1,000 to 1,500 unique separate resources that have been generated every single week. And so what we critically need are better ways to discover and explore this vast amount of data so we can take advantage of it and better understand the virus and the disease. And so to that end, we developed Outbreak.info, which it really encompasses 3 separate parts, which I'm going to talk about. So the first is a standardized collection of resources that's searchable. And so I think what's unique about this is that there are a lot of data curation projects that are focused on one particular type of data or one particular type of resources like publications. And this attempts to unite all of these together into one searchable platform so you don't have to go to 12 different tabs to find what you're looking for. And so as of today, there are over 128,000 separate resources, which we have tagged. And this includes things like publications, including preprints, clinical trials, data sets, and protocols. The second portion of Outbreak.info is an explorable interface, which allows you to delve into epidemiology data. And so really to be able to try to understand how the epi data is changing both over time, but also by location. So you can explore data at the regional level, at the national level, but also at the subnational level, so at like the state level, at the US metro level, or US counties. And then recently, we've been working to incorporate real-time mutation surveillance reports, which Christian alluded to at the beginning of the day, which allow you to track variants over time. And so I really want to focus on this and show how the three components, the mutation surveillance reports, the epi data, and the resources all play together. And so again, this is, our idea of, can we actually start to create a platform to surveil in real time SARS-CoV-2 mutations and variants? And this work has been primarily done by Karthik, Al, and Julia, who have been working tirelessly over the past month to try to get this up. And so to begin with, I wanted to start with the first variant of concern, which sort of alerted the community that we needed to pay closer attention to this. And so that's the B117 lineage, which was originally identified in the UK in December, but has since been found basically in all corners of the world. And so what I found really interesting about this data is that, the identification of the lineage was that it wasn't, necessarily actively being looked for, but they found it sort of by accident when they're trying to understand epidemiology curves that were happening. So in early December in Kent, they noticed that despite all their public health measures, they were seeing an increasing amount of incidence, which was troubling to them. And so they went back and looked at the vast amount of genomic data that they had previously been collecting, and were able to identify a lineage, which is a large phylogenetic cluster, which is now known as the B117 lineage. And so if you look at the epi data, so this is data from Outbreak.info, you can see why they started to become concerned. So in the UK around October, they were in the midst of a pretty substantial second wave, which led them to establish a 4-week lockdown, which began on November 5th. And then that caused cases to decrease over time, but then in early December, they started to tick back up. And so we now know that in the intervening months that happened after this, there was a substantial, I guess, third wave that was experienced, which eclipsed either of them, driven largely by the fact that these were being driven by B117 sequences, which have, research has since found that there's an increased transmissibility for spreading from this set of mutations. And as well, since the identification of B117, there have been other lineages like B1351 and P1, which also show, some troubling behavior in terms of infectivity and immune evasion. And so the question is, can we, instead of looking at this retrospectively, start to think about proactively monitoring mutations? It's clear that the virus is going to continue to evolve and acquire new mutations. And so what we really need is a way to be able to track in real time these lineages and any sets of mutations. And so just looking back retrospectively, at all the sequences that have since been identified as B117, it's clear that, the first sequence was identified in September of 2020, but even in November, there were a large number of sequences in the UK that had the same mutations as B117. So the question is, can we develop a tool that helps us look at this with minimal effort? And again, to sort of emphasize the point that we need an automated, real-time, and customizable mutation tracker to track not only the lineages that we know about, but also the ones in the future. These are the types of questions that the Anderson Lab gets inundated with. And they're very simple questions of, can you tell me how many number of cases of a particular variant have been identified worldwide? And so to that end, we've developed customizable mutation reports, which are updated daily and soon multiple times a day, which allow you to look at any mutation or set of mutations over time. And so I'm going to jump out and break into a demonstration of this, which allows you to show some of the details that you can see with the data that we have. So on the mutation situation reports page, you can see that we have a series of curated lineages. So these are variants of concern or of interest that we are tracking. We also have mutation reports, which are specific to a particular mutation. And then at the bottom of the page, you can select any random Pango lineage that you want. And so in this case, I'm going to take a look at B117 and generate a report from that. And so the first thing in the report to notice is this sort of summary box, which are the high-level statistics that we constantly get asked, just basic how many sequences have been identified with a particular lineage or mutation. So you can see that as of yesterday, there are over 66,000 separate sequences that have been identified with B11 lineage. And then you can also compare that to what is occurring in the US or California or any other locations. The other thing that we did, which we think is pretty important, is to be able to show what the characteristic mutations are within the lineage. This information seems pretty basic, but can often be kind of hard to find. And you can zoom in on any gene of interest because in the case of B117, there are lots of mutations that happen to occur in the S gene. And so you can see, the null mutation, there are 2 deletions that occur at the beginning of the spike protein as well. And then you can start to look at how B117 is actually changing over time as well. And so this is the worldwide trace for it, but we can also change locations. So we can add the United Kingdom. We can also add subnational data, so England, if you'd like. And then we can look at its prevalence over time. And so what you clearly see is that in the United Kingdom, B117 in the most recent batch of sequences is by far the most dominant lineage which exists. But even in early December and late November, there was roughly like 10 to 15% of the sequences that were of the samples that had been sequenced already had the B117 mutation. You can also troll and look at explore what's happening geographically. So looking at the prevalence of B117, in this case, across the different components of the United Kingdom. You can look at it across the US and see that Florida in particular seems like it is leading in the prevalence of B117 as well as worldwide. And to give you a more quantitative view from this chloropleth, you can also look at a dot plot of the cumulative prevalence of B117 that have been identified since the identification in the country. And so one of the challenges with this data is that there are a lot of places which have very few sequences. So for instance, the DRC, it looks like it has 100% prevalence of B117, which is probably not the case because they've only sequenced 2 sequences. And so you can also sort it based off of the total number of sequences, total number of samples that have been sequenced in a location. Again, the United Kingdom is leading the way, but they also have a pretty strong cumulative prevalence of B117 that's only been increasing over time. The other thing that popped out to me, which makes sense geographically and culturally, is the prevalence within Ireland. So cumulatively, since it's been identified in Ireland, they have 54% of their sequences that they have sampled have these sets of mutations. And so the great thing is that we can actually look at this epidemiology data and see, try to develop hypotheses about what's exactly happening. So within the epi data, you can look at any trace of cases or deaths over time. So I can look at the United Kingdom and compare it to Ireland. And just for ease, I'm actually going to overlay the graphs. And you can see that both of them had pretty substantial waves in the past few months. We can also normalize it to population to give a more, I guess, accurate view of what's going on. And so you can see that in both the UK and in Ireland, that starting in early December, they experienced very large waves, larger than anything that they had seen previously in the past year. which seems consistent with the hypothesis that B117 has increased transmissibility. So the last component of the mutation report is that we also pull in any sort of publications and resources that are related to B117. And so I'm actually going to pull this out into the search page so that we can explore it more deeply. And the thing that I really want to emphasize is that this is not only publications, but it's also other types of resources or information that might be interesting or relevant. So for instance, you can filter this by protocols, and you can see that there are 2 protocols which are related to B117, including one from the Grubel lab, which is looking at qPCR screens for identifying variants of concern. You can also do more complicated queries on top of this. So if I'm interested in B117, and in particular interested in the United States, I can pull out the records which are only related to that, and we can see that there are four different resources which are related to B117 in the US, including this paper, which Karthik is going to briefly talk about in a couple hours, but if he doesn't get to it, you should ask him about it in happy hour, about the rapid transmission of B117 in the US. And again, since Outbreak.info harvests and harmonizes all this data together, you can actually go and then explore the publication on medRxiv as it exists. Yeah, and so this is this is a picture of B117, but to be honest, this is a lineage which is pretty well established and a lot is known about it. And so the real question is, what can we do about mutations that, before they're actually classified formally as a lineage, or any arbitrary group of mutations where we want to look at, for instance, things like convergent evolution. And so now I'm going to demo a portion which is allowing you to create any customized mutation reports on any set of mutations, which we hope to release in the next week or so. And so again, you can specify any mutation. I'm actually going to look at the Q677 mutation and have it be mutated to H. So if I add it, I can then create a report based off of it. And the reason why this is interesting is that there's a recent paper from Emma Hodcroft, which is looking at this location, the Q77 position, and noticing that in a number of different lineages, there seems to be convergent evolution where it's existing over time. And so the one change compared to lineage reports is that for a particular mutation, we also show the breakdown by Pango lineage. And so you can see in this particular one, B1525, 100% of the sequences in that lineage actually have this mutation compared to some of the other ones. And I think what's most striking when I was looking at this data is just how widespread Q677H mutation is around the world, even if its overall prevalence hasn't increased too much so far. And you can actually see that if you look at the prevalence over time. In the US, it seems like perhaps there's an increase with this particular mutation. And so that's something that we're keeping our eyes on to see how this is changing over time. Again, all the same functionality exists, so you can look at its distribution across the US. I also learned when I was preparing this that Alabama is sequencing a surprising number of samples. I don't know. I was expecting California and Florida and Massachusetts to be leading the way, but Alabama's holding their own as well. And they seem to have a large number of this particular mutant as well. And then the other part of this is, again, you can go back and explore, the B1525 lineage, which is particularly interesting because it shares a lot of the sort of scary mutations that pop up in other lineages. So for instance, the 484 EEK mutation is present in the B351, which is the South African lineage, as well as the Brazil lineage P1. And then it also shares a number of the deletions that are in common with B117. The last feature I want to show here is also you're able to combine lineages and then add mutations on top of them. And so if we're interested in B117 and, for instance, it's acquiring this EEK, this E384K mutation, we can create a report which is specific to that. So on the background of B117, how many samples have actually acquired this? And so worldwide so far, there are 32 sequences which have been identified only in the United Kingdom at this point, which have this sort of strange mix of B117 as well as the EEK mutation. So that was a very brief overview of the mutation reports and the integration of both the resources and the epidemiology side of Outbreak.info. The last feature I wanted to mention is that everything that we have is both copyable and downloadable. So you can see that there are these little icons at the bottom of every single visualization, which allow you to copy the data and share it, or also to download either the image or the raw data themselves so that you can play with it. So we've spent a lot of time trying to assemble together all of this data, and we want to make sure that other people can take advantage of it too. So please play with it, use our data, and let us know where we can make changes. So to just summarize the capabilities of Outbreak.info, it's really a standardized, searchable, interactive, real-time data exploration system. And again, this encompasses 3 parts, from exploring different types of resources, to looking at epidata, to looking at how mutations and variants have been changing over time. And so I wanted to sort of wrap up by talking about really the unglamorous side of all this process, which makes it possible. And so really the magic between the harvesting and harmonization that allows us to combine data and to be able to view it in a single site. And so this is largely the work of Jinzhou Cheng and Marco Cano, as well as some others on our team, really trying to standardize the data that we are acquiring from a number of different sources. And so particularly on the resources side, standardization and harmonization is a big problem. And this is a problem with heterogeneous data sources, so different types from clinical trials to publications. They don't always agree with each other. But even within a given type of data, they often don't actually talk about things in the same way. And to show why this is a problem, this is a very simple example looking at clinical trial data that we are harvesting from clinicaltrials.gov as well as the WHO. And so both of them have an option to specify the eligibility for a clinical trial. And you can see that the way that they talk about things is slightly different. And so this makes it difficult if you're trying to search, for instance, for any clinical trials for adults, because you basically have to write 2 separate queries if you're asking about the data from clinical trials or from the WHO. And this problem becomes exponentially more difficult the more types of sources that you're trying to integrate. At this point, I think we're integrating, close to 20 separate data sources. And so it becomes a huge challenge. And so what we've done is we've actually developed a standardized schema, which allows these resources to talk to each other and standardizes their output so that instead of having to search separately from each of these data repositories, you can search uniformly across them and find, in this case, clinical trials for adults that are for both males and females. And the real power behind this is that it's built off of schema.org, which is a widely adopted standard, which has been created by Google, Microsoft, Yahoo, and Yandex. And so this means that our data should be interoperable with other forms of data, as well as the fact that we expose all of our metadata for other web crawlers so that things like Google Dataset Search can actually find the data that we have harvested and expose it to make it more easily findable. And so I wanted to talk a little bit more about standardization, and in particular, I'll be the first to admit that I actually really dislike schemas. I find them annoying and quite a lot of work to put together and to deal with. I would much rather spend time thinking about the contents of the data rather than the structure of the data. But how I think about it is it's really like a mise en place for chefs. And so if you're unfamiliar with this term or you haven't watched every season of Top Chef, mise en place is the process by which chefs and bakers actually assemble together all the tools and the ingredients that they need before they combine them together to make their dish. And so there's a lovely quote from Joanne Chang, who owns Flour Bakery in Boston, which I think is really applicable to how we often think about data. We often think that we can just get away with it and sort of gather as we go, and that, oh, we'll measure out the milk when we need it and we'll deal with it when the time comes. But the problem with that is that more often than not, you'll get to the middle of something and you'll figure out you don't have eggs, or that the butter needs to be softened before you cream it. And so I think this is really analogous to the challenges that we often face with data of sort of putting off the standardization until the end of the project, and that it's a lot easier to get all of this in place before you start, before you encounter these predicaments of trying to figure out how to integrate data together with other sources. And so to that end, we've been working with the NIAID Data Dissemination Working Group to develop a metadata standard for data sets that should describe all infectious disease data sets. And so the idea behind this is to develop a common uniform base schema with a minimal set of terms built off of schema.org to promote interoperability, but also adding in some things that we think are essential to help describe infectious disease data sets, such as what's the disease that you're actually trying to study, and what's the host species that the data set covers. And again, the schema is meant as a base so that you can also extend it to add in things that are specific to your particular data set. And the whole point behind this is that metadata standardization, especially of data sets, allows people to find your data so that instead of having to search for a number of different things, they can search unified fields across a number of different data providers. And then the second more tricky issue with standardization is within the data itself. And by doing so, this really allows people to use your data. And so this is an example of what Al has been fighting with over the past week of trying to normalize name locations across the GISAID sequences. And so at the far right, you'll see a very zoomed out image of his code, which is a lot of just making sure that names agree. And this is extremely boring work, but it's essential so that we can make sure that sequences that are labeled Canary Islands and sequences labeled Canary_islands are both identified as the same place. And so this standardization really allows us to take better advantage of our data and to spend less time dealing with cleanup and obnoxious issues like that. And so I wanted to end with the idea that good structured data really allows you to ask tricky questions. So none of what we're able to do would have been possible without dealing with harvesting and harmonization of all these different data sources. And so the more that we can do this from the upfront, the easier that becomes. And so I wanted to end with a brief overview of where we're going next, in particular with mutation reports. So first, the custom mutation reports that I demoed this week will be hopefully released next week. And so this will allow you to ask questions about what about a particular lineage with some random mutations added to it? Or what about one particular mutation that is of interest? The second thing that we're working on is developing custom location reports. So right now, everything is really based around exploring a particular mutation, but you can flip the question and ask what lineages and mutations are predominant in a particular place and how are those changing? Similarly, one of the things that we also want to add is lineage comparisons. So answering very basic things like how does B1351 compare to B117? But then also how are they changing? How are they changing over time? But also what is their geographic distribution? And the more tricky but interesting question is, trying to identify from the start what is abnormal expansion. And so the real question is, can we start to think about, if we were looking back in November at the UK data, can we identify B117 as it's starting to pick up? rather than, a few months later. And then the last thing is that we want to go to county level data for the US, where we have enough data to be able to answer questions like what the California variant B1429 is doing in San Diego. And the last thing I want to mention is that, we're really interested in trying to make this a tool that everyone can use. So if you have suggestions on either problems that you see or functionality that you would like to add, there's a link to GitHub to be able to file an issue and suggest something that we should do. So lastly, I wanted to end by thanking the entire team. We have a really diverse and collaborative group of people with a lot of different expertise that I think melts together in a really remarkable way with Outbreak.info. I wanted to particularly thank the funding sources, and especially NIAID and the OGAC team, because they have been incredibly supportive over the past year and also have had a number of really interesting suggestions and questions to ask about our work. And then the last thing I want to thank is the data providers. So aside from the people in the Anderson lab who are madly sequencing, lots of samples, we don't actually generate any of these data. And so we're just kind of data sponges that suck it up from various places, and that really allows us to do all this work. And so in particular, the epi data is generated from data from Johns Hopkins and the New York Times. The genomics data comes from GISAID, and then on the resources side, there are about 14 different data sources that we're pulling from to assemble this harmonized, harvested collection of resources. So I'll end there, and I'll allow anyone to ask any questions.
Awesome. Thanks very much, Laura. That was very good. We have time for, let's do one question if we have a question.
Yeah. Hi, Laura. Stephen Walensky. You being an editor of the HIV Lano database, I appreciate everything you're going through and such. So you have a lovely geographic search interface, but do you have a search interface for the sequences individually, similar to what we did with NextStrain and the HIV database? Do you have a browser that lets us look at both the genome and the proteome? And one of the problems we had with the HIV database was knowing the integrity of the samples that we're getting and how good the sequences really were, and the different platforms in which they were attained, some of which for nanopore sequencing obviously had more noise in it than other platforms as well. So, and lastly, will you have interface for any tools for working with the sequences that'll allow you to directly do it? I know that's not one question, but sorry.
That's a lot of questions, and let me make sure that I hit them all, so feel free to follow up afterwards. Yeah, so in terms of looking at specific, lineages or sequences, with the mutation reports, right now you can look for any set of custom lineages. So any newly developing pango lineages like B1525, you can look at how those are changing over time in space. On the genome and proteome question, that's something that we haven't looked into a lot, but I think that's an interesting opportunity to explore in the future. On the quality of data, that's obviously a huge factor, and I think that that's something that we struggle with a lot. I don't know if Al or Karthik have any particular experience or answer to whether or not we're filtering out any low quality sequences. I'll let them answer first.
Yeah, we're actually not doing that step right now. Mainly, one of the factors there is the GISAID's terms of agreement. So we don't actually get we don't actually show like, the individual genomes or the individual mutations. We only look at aggregate numbers. GISAID itself has its own set of quality filters to accept only full-length genomes and things like that. But we haven't added any specific filters on the genomes yet.
Yeah, so maybe I think because we need to move on to the next talk, so let's because some of these are actually big conversations that I think is really important because the concerns of data quality.
Sorry about that. I didn't.
Because I'm with you, I am very concerned about that. not for pangolin lineage calling, so it shouldn't affect the variant reports too much actually, so I'm not too concerned about that. But generally speaking, data quality is definitely a concern in the SARS-CoV-2 world.
Especially just with the number of sequences that exist for a particular place. I think that's a huge thing we deal with. I'll just briefly answer your last question in terms of tools and resources integration. So at this point, we're at the status of being able to catalog metadata about these tools and resources so that you can search and find them, but they're not necessarily integrated within the platform. And I think that's an interesting level of opportunity, but I think we want to focus really deeply on one particular use case rather than trying to solve, add in every single tool that could be used.